Hi, I'm Skylar DiMartino,
I'm Erin Lincoln.
I'm Sophia Beland.
and this is our C phages project, a bold hypothesis for Shygu2. Shygu2 was isolated from an enriched soil sample by 2 students here at Merrimack College. The sample was taken at the coordinates shown on the screen, right next to Mendel Pond, which is in front of our science and engineering building in September of 2021. The DNA was sequenced at Pittsburgh Bacteriophage Institute, and it was annotated using PCAN. The CDD was used to identify primase domain homologies. Shygu2 is a mycobacteriophage isolated on the Mycobacterium smegmatis host. It has 51,410 base pairs and contains 87 genes, with 32 genes in the typical first half forward and second half reverse with 55 genes.
Gene 55 of Shygu2 shares domain homology with bacteria DNA G, while gene 56 contains a zinc finger DNA binding domain. The DNA binding or the zinc finger is shown at the top left of the figure. These 2 primase genes share a significant overlap of 74 base pairs, as shown as the top right of the figure. Because the literature shows no evidence of multiple mRNAs, translational frame shifts, or inteins, we hypothesize that genes 55 and 56 produce 2 distinct polypeptides that interact to form a multi-subunit primase. This occurrence of 2 distinct polypeptides is shown in archaea and eukaryotes and is depicted across the bottom half of the figure. In terms of next steps, further wet lab experiments should be done to determine whether the DNA primase in Shygu2 is made up of 1 or 2 polypeptides.
So the first takeaway that we have from examining Shygu2 bacteriophage is that it is a temperate phage identified as Mycobacterium smegmatis, and it has a 51,410 base pair genome and is part of the A4 subcluster, along with Morpher 26, Cataly 136, Bumblebee 11, CC, and Abby's Ranger, Shygu2's closest relatives. As seen in the diagram here, the family rater comparison is shown of Shygu2 and Morpher, the closest relative. An additional evidence that Shygu2 is part of the A4 subcluster is the characteristic immunity repressor being located far on the right arm, rather than next to the serine integrase. Also, 3 minor tail proteins are found at the left end of the genome between the terminase, gene 2, and lysine A, gene 8, characteristic of cluster A4 phages like Morpher 26 and Cataly 136.